Hello, thank you for clicking this video. For today, we will be talking about the Kreb cycle. It was made by a German chemist Hans Adolf Kreb. His discovery of this cycle in 1937 marked a milestone in biochemistry. Now, let's dig in! The two molecules of acetyl coenzyme A, made during pyruvate oxidation, will now enter the citric acid cycle, also known as the Kreb cycle, the tricarboxylic acid cycle, or the TCA. The citric acid cycle is cyclical because oxaloacetate or oxaloacetic acid is a reactant in the first step of the cycle as well as the product of the final step of the cycle. Oxaloacetate, acetyl-CoA, and a molecule of water enter the first enzyme in the cycle, called citrate synthase. Then the water hydrolyzes acetyl-CoA, causing the CoA group to break off and leave the enzyme. Oxaloacetate and the remaining acetyl group join together to create a molecule called citrate. And then, the newly formed 6-carbon molecule leaves the enzyme. Now, citrate enters the aconitase enzyme to be rearranged into isocitrate. Isocitrate enters the isocitrate dehydrogenase enzyme to be decarboxylated. The CO2 molecule diffuses out of the cell as waste. NAD positive enters an active site on the enzyme. On the next step, NAD positive is reduced by a hydrogen atom and leaves the enzyme, accompanied by another hydrogen proton. Then, the remaining alpha-ketoglutarate molecule leaves the enzyme. Now, the molecules of CoA, NAD positive, and alpha-ketoglutarate enters active sites on the alpha-ketoglutarate dehydrogenase enzyme. NAD positive is now reduced by a hydrogen atom and leaves the enzyme, accompanied by another hydrogen proton. The instability caused by this NAD-positive reduction helps initiate the decarboxylation of alpha-ketoglutarate. Thereafter, the CO2 molecule diffuses out of the cell as waste. Now, none of the carbon atoms from the original glucose molecule remain. The CoA molecule replaces the missing carboxyl group to create a molecule of succinyl-CoA. Then, the succinyl-CoA enters the succinyl-CoA synthase enzyme. The succinyl-CoA is now reduced by a molecule of water. In the process, an inorganic phosphate enters an active site on the enzyme and displaces CoA. CoA is released and can be used again in other reactions. A molecule of guanosine diphosphate enters the enzyme. The phosphate group is transferred to GDP, forming guanosine triphosphate. Succinyl-CoA turns into the molecule succinate after losing the phosphate group. Then, an ADP enters an active site on the enzyme. The phosphate group leaves the G3P and joins the ADP to create a molecule of adenosine triphosphate or ATP. This method of making ATP is called substrate-level phosphorylation. Phosphorylation means to add a phosphate group. Substrate level means that this reaction happens at the active site of an enzyme. The GDP, ATP, and succinate now leaves the enzyme. The succinate dehydrogenase is now considered to be a component of both the Kreb cycle and the ETC. It can be found embedded in their inner mitochondrial membrane with other components of the electron transport chain. The succinate will now enter an active site on the succinic dehydrogenase enzyme, which has a molecule of flavin adenine dinucleotide or FAD embedded within it. In this step, FAD is a cofactor. A cofactor is a non-protein chemical compound that helps biochemical reactions take place. FAD is reduced by 2 hydrogen atoms from succinate to form FADH2. See the illustration for example. The reduced flavin adenine dinucleotide will subsequently be used by electron transport chain, and the newly created fumarate molecule leaves the enzyme. Fumarate and a molecule of water will now enter the fumarase enzyme. The water molecule splits into one hydrogen atom and one hydroxyl group that will join fumarate, thereby turning it into a molecule of malate. This malate enters the eighth and final enzyme in the cycle, malate dehydrogenase, wherein NAD positive enters an active site on the enzyme. The NAD positive is reduced by a hydrogen atom and leaves the enzyme accompanied by another hydrogen proton. The malate molecule has now been converted into oxaloacetate. This oxaloacetate leaves the enzyme and is free to join with another molecule of acetyl-CoA and repeat the cycle. This cycle happens to each of the two acetyl-CoA molecules made during the pyruvate oxidation. This equation shows the complete chemical reaction for each molecule of acetyl-CoA that goes through the citric acid cycle. These products in the illustration will now have a corresponding variety of destinations. I do hope that learning the Kreb cycle, commonly known as the citric acid cycle, has been fun. This has been Sophia Lalaine Camasura of BSN1E saying thank you and have a good day. Mabuhay!